All right, hey AP, I'm in a room in the library, so there's going to be a little bit of an echo with this, but it's the best place and to do this. Okay, so here's the questions I gave you just to review for cell signaling. I'm just going to kind of go through them with you. Okay, what I suggest is you going through them first, or at least pausing before I go over the answer and trying to figure out what you think the answer is, and then hearing my response to what the right answer is. Okay. For time, I'll probably skip over some of this stuff, but nonetheless, let's go ahead and get started. So this first one is a short response question, and this is a great short response question. Really, really, really great. Something that looks a lot like stuff that would be on May test. And so solid tumors, so this is, you obviously have your reading, right? And so solid tumors are clusters of cancer cells and contain blood vessels. Molecule B, okay, they often don't give names. Sometimes they just randomly say, letters, okay? Because again, we're learning concepts. To the wild type BREC protein, okay? In the plasma membrane of certain solid tumor cancer cells, see figure 1, the cancer cells express the membrane protein A and sometimes stimulate increased growth of blood vessels into the tumors. Okay, so here's what figure 1, this is figure 1, right? And so figure 1 is talking about when B binds, we have this like transduction, and we have active and inactive and active and inactive, and then finally you stimulate growth of blood vessels into tumors, which of course feeds them and gives them nutrients and all that sort of stuff. Okay, cells with a particular mutation, that's an important word to think about, in the BREC gene, have much increased expression levels of A and stimulate greater growth of blood vessels than do cancer cells with the wild type. Again, the wild type is a fancy way of saying like just the most, common, like common in a population, okay? The cells with a mutant BREC can trigger intracellular signaling in the absence, that's a huge clue right there, in the absence of B, okay? Researchers propose that the signaling pathway modeled in figure 1 is triggered by activation of the wild type BREC and associated with phosphorylation, went over that in my video, and activation of kinase, gotta know what that word means, D, expression of A, and the ability of cancer cells to stimulate organ, blood vessel growth, and to, at an organismal level. Okay, so here we go. Gotta understand this. Of course, this is, looking at this video, need to be able to understand what's going on with it. So here is B, and here is kinase, and here is the response, right? So in Seidel signaling cascade, you always have your signal or reception, and then you have your transduction. Sorry, Apple Pen not working, still not working. Transduction, and then response. Okay, so let's go through the questions again. Again, definitely pause before you dive into this. Go through and answer them yourself, and then come back and see what I have gone over with you. Okay, so letter A. Based on the signaling model, describe the role of molecule B. Well, molecule B is the ligand, and it binds to the receptor. Okay, one sentence, done. Explain how the addition of a phosphate group to certain amino acids of BREC lightly affects the tertiary structure and function of BREC. Explain how the addition of a small RNA could prevent the activation of kinase D. Okay, well, adding a phosphate, which has a charge, okay, certain groups of that, sorry, I think I'm like right beside a door opening and closing. So sorry, that might be a little annoying through the video, but nonetheless, we're gonna keep moving forward. So explain how the addition, so that's a charge, right? So this is all about shape leads to function. Shape leads to function. And so it's going to change the tertiary structure and thereby affect it. Okay, not expecting you to know this part yet. We haven't covered like microRNA or anything like that in translation and degradation, any of that. I'm not expecting you to know that yet. Okay, based on the proposed signaling pathway, predict, okay, so that's your action verb, predict the relative amount of phosphorylated to unphosphorylated kinase D in the Breck wild type cells when the cells are grown in nutrient broth lacking B, and justify. Okay, predict and justify. You'll definitely see this in May. And so predict and justify. First off, phosphorylate. So always go back up to what they give you, right? They're asking about phosphorylated kinase D to unphosphorylated kinase D in the absence. Okay, well, in the presence of B, we're making the phosphorylated. Okay, so in the absence of B, there will be way more unphosphorylated kinase, because again, when it is phosphorylated, that is in response to B being around. Okay, just by your prediction, wild type Breck is inactive. Okay, so this protein is inactive, okay, until B binds to it. Okay, so you have to have B around for this to become active, and thereby doing all of this and then eventually getting to this. Okay, so wild type Breck is inactive in the absence of B. The receptor will not stimulate intracellular signaling or the signal transduction pathway, and thereby the phosphorylated kinase will not be made, thereby the kinase will remain unphosphorylated. Okay, and then letter D, cells are treated with a compound that prevents the inactivation of kinase D. Prevents the inactivation of kinase D. Okay. Researchers claim that Breck wild-type cells treated with this compound will stimulate blood vessel-forming cell division to the same degree as Breck mutant cells do in the absence of B. Based on the information provided, provide reasoning to support their claim. Okay? Both cells will now have a continuously active kinase D, all right, which is right here. Okay? Both of them will have an active kinase D, okay? Because kinase D now, since it's prevented from being inactivated, it's always on, okay? Researchers' claim is correct because since kinase D is always on, it will lead to the phosphorylation of this kinase and then thereby creating the response, okay? So this part of the cell signal transduction pathway is always on, thereby everything afterwards continues, okay? And so the continuous expression of A will stimulate blood vessel forming cell division by both types of cells, okay? So that's just one example of like a longer response question, okay? Just because I'm not sure how long this video will get, I'm going to go to multiple choice now because I have a feeling this might be hard to upload into YouTube the longer it is. And so I might just post the answers to this one in like a document for you to look at. But nonetheless, here we go. All right, the epinephrine signaling pathway plays a role in regulating glucose homeostasis, and muscle cell signaling pathway is activated by the binding of epinephrine to the beta-2 adrenergic receptor. A simplified model of the pathway is shown. Simplified, okay, it's a model, trying to understand what's going on. Here's epinephrine, that's the ligand. Here's beta-2 receptor. There's G protein, super important, we talked about that. Here's adenylyl cyclase. There's CAMP, which is the secondary messenger. Okay, protein kinase A, phosphorylase kinase, glycogen phosphorylase, and then glucose 1, which leads to glycolysis, which is a process of using glucose to make convert the energy in glucose into ATP energy, okay? So I'm going to let you kind of pause the video here, read through these two questions, and I'll highlight the answer after you're done pausing. All right, so in pause. All right, so for number 3, based on figure 1, which of the following statements best describes epinephrine signaling pathway? The answer is B right here. It involves enzymes activating other enzymes. right? Kinase activating phosphorylase kinase. Number 4, which of the following statements best describes the role of adenyl cyclase in the epinephrine pathway? It accelerates the production of the second messenger, which we kind of already labeled, okay? Number 5, which of the following outcomes will most likely result from the irreversible binding of GDP to the G protein? Irreversible binding. Irreversible. Can't undo it. And if you notice up here, you have to have GTP come in, okay? And so what will happen? Glycogen will increase because the pathway will have shut down because GTP can't bind. A researcher claims, that the epinephrine signaling pathway controls a catabolic, that's an interesting word we talked about, in muscle cells. Which of the following statements best helps justify, good keyword there to underline, the researcher's claim? Okay, pause. And then the best answer is this answer right here. Glycogen phosphorylase catalyzes the breakdown of glycogen, which is a catabolic process. Okay, glycogen to glucose is breaking it down from a polymer to a monomer. Number 7, not relating to the image. Ethylene causes fruit to ripen. Signaling pathway receptors activate transcription factors, which ultimately leads to ripening. We haven't talked about this yet, but this is just factors needing to express. This is all about gene expression. All right, gene expression. Making proteins from genes. Which of the following best supports the claim that ethylene begins the signal transduction pathway that leads to the ripening of fruit? Which of the following best supports it? Okay, from a scientific perspective, the best answer is this. Because if these receptors are changed, which means they lose their functions, they can't bind to ethylene, ripening also changes. So this is a direct correlation, cause and effect. Number 8, glucocorticoids are steroid hormones that control cellular responses through several different signaling pathways. One of the signaling pathways involves the glucocorticoid receptor, an intracellular protein that's activated. Intracellular is an interesting term. That's activated by binding to the glucocorticoid molecule. Simplified molecule shown below. Okay, here we go. Again, I would always label it ligand, receptor. It's inside the cell. Accessory proteins around. Activated receptor, because why? The ligand has bound to the receptor. It goes into the nucleus, and this is the response because of changes in gene expression, okay? Which of the following statements best predicts the effect of a mutation that results in the loss? Mutation that results in the loss of the glucocorticoid receptor's binding function. If you study the picture, when it binds, these are removed, and thereby, this is the answer now. If it no longer binds, the accessory proteins will stay attached. Number 9. Vertebrate responses involve communication over long and short distances. Which of the following statements best helps explain how cell surface proteins, and T cell receptors mediate cell communication over short distances? All about short. Okay. This is long. That's long. Directly is a good word. So that seems to be right. Let's make sure about D. The protein's binding molecules secreted. Nope, that's endocrine. Okay, so the answer is C. Number 10. Signal transduction pathway. Here we go. Activated molecule, blah, 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 blah. A DNA mutation. Look how many mutations they bring up. They'll do the same in May, right? This helps us all see. Do you know the concepts? You know how it works. Do you know what would happen if it's not working correctly or if something's changed, okay? A DNA mutation changes the shape of the extracellular domain of transmembrane receptor protein A. It changes this guy's shape. Which of the following predictions is the most likely consequence of the mutation? 2 and 3 can't continue without 1 being around. The molecule that normally binds to A will no longer attach deactivating. That is the right answer. B. Okay. 11. Notch is a receptor protein displayed on the surface of certain cells in a developing fruit fly embryo. Notch's ligand is a membrane bound protein called delta. Okay, the ligand is called delta. That's displayed on the surface. When Notch is activated by its ligand, the intracellular tail becomes separated from the rest of the protein. This allows the intracellular to move into the nucleus and alter expression. Again, we're reading different books. This is DNA of genes, DNA to protein, transcription and translation. Which of the following statements best explains delta's role in regulating cell communication through the Notch signaling pathway? Well, delta, okay, if you read through it, pause, of course. And the answer is delta restricts communication to short distances within a developing embryo. How do we know it's short distances? Because it says the word adjacent. 12. In flowering plants, plasma desmata are narrow channels through which connect the cytoplasm of adjacent cells. An explanation of how plant cells communicate across cell walls will most likely refer to the diffusion through plasma desmata. Which of the following? What's diffusing? And the answer is this. Nothing else would be meaningful or small enough to go through. In a certain signal transduction pathway, the binding of an extracellular molecule to the cell's surface protein results in a rapid increase of CAMP. Okay. The cyclic AMP binds to and activates cytoplasmic enzymes that activate other enzymes. Which of the following statements best describes the role of CAMP in the signal transduction pathway? This is a straight definition, kind of second messenger. And then finally, last one, number 14. Hope this was helpful to you guys. A student claims that the Y chromosome contains a sex-determining region gene known as SRY, which causes male fetuses to develop testes. Which of the following provides correct information about cell signaling, well, that's a key word, that supports the claim? Cell signaling. Well, signaling is all about three different things, right? And so you have to be able to bind. That's a good word. Deleting the portion of the chromosome, that would not be based upon cell signaling. RNA would be, that's all, again, translation. And then that's anatomy in letter D. So the answer is A, because it says binds to specific regions. Remember, binding, right? The signal binds. And then we have a signal transduction cascade, and then we get a response. Hope this was helpful. Yeah, let me know. Keep working hard.